The world is changed. I feel it in the water. I feel it in the earth. I smell it in the air. Much that once was is lost, for none now live who remember it.
Prexy's Pond, a seemingly pleasant Concordia locale. But is this the truth? What is the truth? The truth is spirit, but what is spirit? The spirit is the self, but what is the self? The self is a relation which relates to it.
Lie. I thought we said no Kierkegaard today.
Sorry.
Prexy's Pond is home to many things. Ducks! Japanese horror flick creatures, things from Scotland, naked college graduates in golf carts, but worst of all,
bacteria!
Two brave Concordia students have taken it upon themselves to investigate this disturbing reality.
We were inspired from a previous experiment, one where we tested for bacteria from Deer Lake in northern Minnesota. Digging 2 inches beneath the surface of the lake bottom, we gathered a handful of soil sample. After drying the soil and diluting it in a variety of strengths, we incubated the samples with nutrient agar.
To our dismay, very little or no bacteria grew in the petri dishes. We had to ask ourselves, why? Since the soil came from a place where bacteria would constantly be surrounded by liquid water, we hypothesized that the drying process killed off the indigenous bacterial life.
But we came up with an ingenious scheme to confirm our idea.
Well, ingenious could be contested.
Having decided to test soil from Prexy's Pond, we still needed to plan our next steps. Our new hypothesis read thus. Some or all of the bacteria in Prexy's Pond are dependent upon a liquid water environment for survival. Longer periods of drying will result in a greater rate of bacterial termination. Shorter periods of drying will result in a lesser rate of bacterial termination. Our plan was to gather a bunch of soil from the base of the pond and then divide them into 4 equal parts. Next, we placed on their aluminum covers 1 post-it each labeled with the following designated drying times: 36 hours, 24 hours, 12 hours, and 0 hours.
Since our in-lab time started at 10:30 a.m., we decided that we should start the drying at 10:30 p.m. 2 days prior to the actual lab date. When 10:30 p.m. on the night of November 7th rolled around, we uncovered the amount labeled 36 hours. The following morning at 10:30, we uncovered the amount labeled 24 hours, and that night at 10:30, the amount labeled 12 hours.
We would then proceed with diluting our soil samples into 4 water blanks via pipette transfers. We would place small amounts of these dilutions into 4 petri plates per sample at differing dilutions.
Having completed this, we would then place 20 milliliters of nutrient agar into each petri dish, plus 20 milliliters of agar into a blank petri dish, which would serve as our control, much like the process used in the original soil bacteria lab.
All we'd have to do then is incubate them.
At 37 degrees Celsius.
Once again, this should...
Is he familiar?
Once again, this should seem very familiar to you.
The discrepancies between the expected results and the actual results, in fact they were stark opposites, can be explained by one of 2 possibilities.
Firstly, we may not have properly or sufficiently sacrificed to appease the bacterial gods. Seriously, I mean we sacrifice $350 every month to the rent gods, 333 minutes a week to the life and universe class gods, and 4 hours a week to the band gods. Don't even get me started on the homework gods, Peter Schultz.
But we have adequately planned and given passion and energy to this process, which would only leave one last explanation.
Sabotage! Sabotage!
Casey! Casey!
It's
Casey!
sabotage!
It's sabotage! Casey!
No, guys. Oh, it's not sabotage.
We went back to the incubator on Thursday, November 11th to check our petri dishes for bacteria colonies and gather the data. Our results, we found, were disturbingly counterintuitive within the context of our hypothesis.
As you can see from our chart, the longer the soil had been drying, the more bacteria was yielded. We also noticed that the expected range of bacteria, somewhere between 30 and 300 colonies, was only obtained in one petri dish. Our control remained clean, indicating that our process adequately prevented contamination.
The petri dishes with the 10 to the negative 4 dilutions, which were most probable to house the 30 to 300 range of colonies, were rendered unreadable through an unfortunate error in the laboratory that previous Tuesday.
While transferring sample dilutions into water blanks, we unwittingly used 0.1 milliliter pipettes instead of the intended 1 milliliter pipettes, making each of our dilutions a tenth more diluted. As a result of fancy math by Dr. Pratt, we were able to salvage these hyperdilutions by changing the amounts added to the petri dishes.
However, in order to obtain a 10 to the negative 4 dilution in a petri dish, as our water blank intended for this dilution now represented a 10 to the negative 5 dilution, we were forced to add an entire 10 milliliters of this liquid into the petri dish, which would then also house the 20 milliliters of nutrient agar, a total of 30 milliliters of fluid in one petri dish.
This proved to be too much liquid to set in the incubator, resulting in a clumpy, ugly mess of brown something. In these dishes, some things might have grown, but it was impossible and unwise to count and use this data. In other words, the agar wouldn't set.
Something else that may have inversely influenced our bacterial growth was that this soil was primarily black dirt. When we had performed the soil bacteria experiment earlier this semester, the activity that led us to use this latest hypothesis, we had used a clay-based soil sample.
In that experiment, we noticed one other student group with a clay sample having difficulty growing colonies. It may be that clay is somehow structured or composed in such a way as to stunt bacterial growth.
The pond itself was extremely cold. Sticking my hand into it proved a very unpleasant experience. The longer a sample is uncovered, however, the more opportunity it has to warm up. As these samples are warming over their designated drying times, heat could have brought about chemical reactions, making for a conducive environment for bacterial growth.
Our final guess as to why our data turned out backwards is that when we uncovered the soil samples in the kitchen, we allowed opportunity for foreign bacteria, aka kitchen scum, to invade and manifest in our soil samples.
So, to recap, there are 4 reasons that might have contributed to our backwards results. 1. Our mistakes in the laboratory have irretrievably shifted our experiment. 2. Dirt is more conducive to growing bacteria than clay. 3. The warming that took place with the uncovered samples brought about increased chemical reaction, allowing faster bacterial growth. And finally, the uncovering of our samples in our kitchen allowed foreign bacteria to contaminate the soil. As Dr. Pratt mentioned to us, when she heard about this experiment, she was told that the drier the soil sample, the better. This may be true. However, a single experiment cannot rule out a well-founded hypothesis. Furthermore, future experimental questions may be asked to build upon our project. If we were to continue experimenting, we would want to do so in more of a community, gathering other like-minded scientists in order to provide a larger data set and a mechanism for preventing human error.
Soil samples of varying composition should be tested in the same fashion to see if the fecundity of bacteria relies upon the type of soil gathered and its relationship to liquid water. Uncovered samples should be kept in a sterile environment, regulated at the same temperature from whence they were drawn.